Hi, I'm Yusha from School of Environmental Science and Engineering at the Southern University of Science and Technology, Shenzhen, China. Here I will introduce our review article on the strategy and tools in illumina and nanopore-integrated metagenomic analysis of microbiome data. Well, for modern metagenomics, relies heavily on the high-throughput sequencing platforms, and the two platforms we cover in this review is the Illumina short-read sequencing and the Oxford nanopore long-read sequencing. And from these two tables here, we can see the Illumina short-reads and nanopore long-reads are complementary to each other from many perspectives. Therefore, there is a growing trend to integrate these two technologies into the metagenomic investigations. Therefore, in this review, in order to provide a systematic and timely knowledge work for integrating the short reads and long reads in metagenomic microbial data analysis, we explained the workflow of common analytical strategies for the integration and provide in detail about the algorithm foundations and the application practice of the common tools. And plus, we have provided the sample preparation protocols commonly used for the user's reference. For the strategy part, the first strategy is known as the genome-centric strategy, which is based on the de novo assembly of the metagenomic dataset. The target of this genome-centric strategy is to get the high-quality mix of the populations of a community. This can be achieved by two passes. The first one is known as the hybrid assembly, in which the Illumina short reads and nanopore long reads will be hybridly assembled together to get the primary assembly for subsequent analysis. The challenge of this hybrid assembly is the high computational cost. Usually, a lot of RAM and core time are needed to finish this hybrid assembly. So in order to overcome this obstacle, a lot of researchers have using the nanopore alone dataset for their primary assembly. Well, because the errors may persist on the nanopore derived assembly, the assembled context needs to go through 3 steps of polish before use. The first one is the self-correction of long reads itself, and the second one is where Illumina dataset got integrated into the workflow. We use the Illumina short reads to polish the indel errors on the nanopore assembled context. And the third step is the frame shift correction based on the alignment to a well-documented protein database. Well, the challenge for this nanopore assembly is that the nanopore sequencing nowadays is still costly. So for many cases, that the nanopore dataset won't have enough coverage of the community. So the insufficient community coverage would cause bad assembly of this nanopore assembly approach. An alternative strategy is called the assembly-free gene-centric strategy. The core idea here is to study the community functional diversity directly based on the polished nanopore long reads rather than the assembled context. The practical importance of this approach is because that the nanopore long reads are not easy to be assembled, presumably due to the low coverage issue I have mentioned before. Where on the other hand, the nanopore long reads is as long and therefore as informative as the assembly in terms of to link the functional diversity to specific microbial lineage. In order to achieve this, the nanopore long reads just go through the 3 polished steps as I mentioned before, and then can be subject to phylogenetic or functional annotation by different tools. Well, at last, I would like to thank you for your interest in our work, and thank you for your interest in the iMeta journal. Just like us and help us spread this work. Bye-bye.